So we talked in our last video about light and how it's absorbed by the pigments, and in this we're going to have a look at the actual process of photosynthesis and what that light energy is used to do in that process. So to do that, we're going to have a look first of all at your chloroplast and the structure of your chloroplast. So you've got a double membrane that's there, you've got an inner membrane and an outer membrane, and then you've got these here that are called thylakoids, and these are, I'm trying to get my pen to work. So your thylakoids that are here, these are stacked up on each other, and when they're stacked up on each other, they're kind of these disc-like sort of frisbee characters. When they're stacked up on each other, it forms something called a granum and there the photosynthetic pigments are all contained within the grana or granum. And this is the part where the light dependent stage of photosynthesis takes place. Here we've got this fluid filled part inside the chloroplast, and that's called the stroma, and that is where carbon fixation takes place. So let's have a wee look at the process that takes place. First of all, looking at the light dependent stage, or sometimes also called photolysis. Now in the chloroplast, you've got that light energy absorbed by chlorophyll A. What happens is the electrons in the chlorophyll, they become excited by that light energy, and they're raised to a higher energy state. And these high energy, so here we've got our light here we've got our chlorophyll, the light energy there that is absorbed by the chlorophyll, and that makes, it's got this little wiggly bit here to depict high energy electrons raised to a higher energy state. Now those high energy electrons are captured by what is known as the primary electron acceptor. And these electrons are then passed along an electron transport chain. If you remember the process before, where you had the electron transfer chain in respiration, it's the same principle that is here. The electrons are passed along that transport chain, and that pumps hydrogen ions into the grana. So what we're looking at here, this kind of reddish colour that's here, that is depicting the membrane of the grana, and the hydrogen ions end up being pumped through into the inside of the grana. Now, when they're inside there, they build up in this high concentration here, and what happens is the hydrogen ions then pass through and out of the grana, and when they do that, they pass through ATP synthesis. Now you remember that before from your electron transport chain in respiration. So it's the same process here. It's this enzyme that is here, your ATP synthesis, and it is used to make ATP. And that ATP, it then goes into the next stage for the carbon fixation, or sometimes known as a Calvin cycle. Now, as well as that happening, some of the energy is also used to split water. And when that water is split, then you produce oxygen and you produce hydrogen. And what happens is the hydrogen is picked up by a hydrogen acceptor. In this case here is called NADP. You remember when the situation is there in respiration, there's NAD, there's not a P at the end of it. And how I always remember this is, if we're talking about photosynthesis, the hydrogen acceptor has got a P at the end of it for photosynthesis. If you're talking about respiration, there's no P at the end. So the hydrogen is picked up by that hydrogen acceptor, making the NADP, making it into NADPH, and that moves into the carbon fixation part, or the Calvin cycle, the next part of the process. So at the end of this process, you've produced ATP, you've got NADPH, so you've got your hydrogen and your ATP that are going into the next stage. The ATP is being produced by the ATP synthase. The NADPH has been produced by energy that has been absorbed and producing these high energy electrons, and the water is split and you get your hydrogen, which goes on into the next stage. It's been accepted by your NADPH, your hydrogen acceptor. So the next stage is our Calvin cycle. And as I've said, at the end of the first stage of photosynthesis, the light dependent stage, the hydrogen in the form of NADPH and the ATP, they're essential for the second stage known as the Calvin cycle. The oxygen, as we know, just diffuses out of the cell. So the Calvin cycle, or the second stage, it takes place in the stroma of the chloroplast, and we'll look at that process in the next slide. So here's looking at carbon fixation. So first of all, we've got our carbon dioxide here. So that enters into the cycle and it becomes attached to this molecule here, RUBP. That's short for something called ribulose biphosphate. And the reaction is controlled by an enzyme here called rubisco, and that is short for ribulose biphosphate carboxylase oxygenase. But you don't need to know the whole of that name, you just need to know this rubisco term here. So this enzyme catalyzes this reaction or controls this reaction, and the carbon dioxide and the RUBP, controlled by this enzyme rubisco, produce this intermediate compound here. And what happens next is this intermediate compound, hydrogen, it joins with the hydrogen from the NADH, and as well as that, the compound is phosphorylated from the ATP, which supplies the energy. So by phosphorylated, what we mean is an inorganic phosphate is added to it, and we form this substance here called G3P, and that is short for something called glyceraldehyde 3-phosphate. Now some of that G3P is used to regenerate RUBP to continue that process, and then the remainder of it is used to make glucose. So that is the process of photosynthesis. Your light dependent stage, where you produce your ATP and your hydrogen ions that are attached to your hydrogen acceptor, and then the second stage here, the Calvin cycle, where carbon dioxide enters in. The enzyme rubisco controls the reaction where RUBP is and carbon dioxide combine to form an intermediate compound. That intermediate compound is then reduced by the addition of hydrogen and phosphorylated by the addition of an inorganic phosphate to produce the G3P, which part of which goes on to make more RUBP, and some of that goes on to make glucose. You'll notice here as well that some ATP is used up in this process of making the G3P into RUBP. I'll add a photosynthesis video as well for you to watch. It's got some extra information in it talking about CAM or C4 plants that you don't need to know about, but it's got the photosynthesis process in it. So what happens to those sugars or those carbohydrates that are produced by photosynthesis? Well, they can go to the following uses. So for example, glucose that's formed in the plant can be used for cellular respiration, and that can provide the plant with energy for growth or for cell division, reproduction. Some of the other sugar molecules can be made into more complex carbohydrates. For example, they could be built into long chains of cellulose to make your structural carbohydrates that are used to build cell walls. or they might be linked together in long chains and packed into spherical starch grains, such as your storage carbohydrate starch. And other molecules of carbohydrate produced by photosynthesis might be passed to other biosynthetic pathways, and these contribute to the production of various other metabolites, for example, amino acids, proteins, nucleic acids, nucleotides, fats, and oils.